Okay, so we'll look at a new chapter today on nucleic acid and protein synthesis. There's something that we have kind of touched upon a little bit by little bit in the past, in the previous few topics. Okay, so at the core of it, okay, so of course we always talk about in the last chapter we learned about the importance of cell division. Okay, of course, one of the core and most important tasks of cell division is to perform DNA replication. Okay, because your DNA is what we call the hereditary material. Okay, so everything is in there. Okay, so I always like to compare your DNA. Okay, so to your your nucleus to your CPU. Okay, where all the core information that is relevant to a particular process or like a particular organization or house or whatever is stored. Okay, so now we'll start with the structure of DNA and RNA. Okay, so we will talk about DNA. So as you know, DNA stands for deoxyribonucleic acid, and RNA is ribonucleic acid. Later we're going to look at the structure in detail. Of course, just by the name itself, we know that for your DNA, in terms of the nucleotide, okay, they lack one oxygen as compared to compared to the RNA. Okay, so gene. Okay, so gene is a section of DNA. Okay, so maybe you copy this first. Okay, so we always talk about gene. Okay, so gene is essentially a section of a DNA, and if you remember, I think we also, this is not the best definition of gene. Okay, so gene essentially is what we call an information unit. Okay, that encodes a particular polypeptide chain. I think if you want, you may also just put it here. Okay, so basically, okay, so a section of DNA which encodes a polypeptide. Okay, so when we say gene, okay, basically one gene corresponds to one protein. Okay, so hopefully you know that your gene is actually ultimately is an information for synthesizing protein. Okay, and in the last chapter we have already mentioned. Okay, so chromosome essentially is a DNA molecule. Okay, so it is just a very large DNA molecule. Okay. And DNA controls which enzymes and other proteins are synthesized by cells. Okay, later we are going to see how DNA actually function as an information for protein synthesis. But again, it is actually something that's been covered in high school, at least for the SPM and UEC syllabus. Okay, and the DNA can accurately copy itself. Okay, so in the last chapter we have learned that during interphase, okay, DNA gets replicated. And of course, always we talk about mutation. Okay, so mutation actually refers to changes to DNA. Okay, can you please tell me, or maybe just based on what you already know, what are the consequences of a mutation? What could be the potential consequence of DNA mutation? Okay, yeah. So substitution is one kind of mutation. I'm more concerned about what can it cause ultimately. Yeah, potentially, yes, appearance may change. Okay, for example, I'm sure you heard about albinism. Have you heard about albinism? Okay, so people where their body lacks ability to synthesize color pigment. Okay, so all their skin, their hair is very fat. Yes, yes. Okay, so that's due to mutation. Okay, of course, genetic mutation can sometimes also cause a disease. Okay, cancer is itself a disease that's due to mutation. Okay, so there are other many other genetic diseases that occur as a result of DNA mutation. But of course, sometimes mutation can also be beneficial. Okay, for example, if we talk about, let's say, from the perspective of bacteria, sometimes a mutation may make them, being able to escape the attack by the immune system of a human, or maybe it allows them to be resistant to particular antibiotics. Of course, in human case, sometimes there may be some mutation that confers a desirable properties. Okay, so nucleic acid is a term that is even more general than just DNA. Okay, so DNA is a kind of nucleic acid. Okay, so nucleic acid generally forms a genetic material of all organisms. Okay, so there are two types, DNA and RNA. Okay, so of course in human, the information is stored in the form of DNA. Okay, but there are other organisms such as certain virus. Okay, if we consider virus as an organism. Okay, so for the case of virus, okay, virus can be divided into two broad categories: DNA virus and RNA virus. When we call them RNA virus, it means that the genetic material is stored in the form of RNA. So if it's DNA virus, the genetic material is stored in DNA form. Okay, and there are polynucleotides, okay, made of monomers called nucleotides. Okay, so just get the terminologies right. Okay, so nucleic acid is kind of just referring very broadly to DNA or RNA. Okay, so nucleotide is the individual unit, just like your proteins would be nucleic acid. Okay, amino acids are like nucleotides. Okay, so they are the monomers. Okay, so structure of a nucleotide is something that you need to know. Okay. Okay, so nucleotide, so there are three components. Okay, there are three parts which make up a nucleotide, whether it is an RNA nucleotide or DNA nucleotide. Okay, so for DNA nucleotide, okay, or RNA, either one. Okay, so always they have a phosphate group. Okay, so they would have a phosphate group and a pentose sugar 5-carbon compound, a 5-carbon sugar. Okay, so you see a typical pentagonal shape, but just do note that, it's not all carbon at the 5 corners. Okay, so one of it is O, but there's the 5th carbon here. So 1, 2, 3, 4. This is a 5th carbon. Okay, and then there's what we call a base. Okay, so later we are going to see there are 4 kind of bases. Okay, so whether it is DNA or RNA. Okay, so this is where they differ, where each of the nucleotide differ from each other. Okay, so of course I have to say that not all so there are other form of nucleotides. Okay, but for genetic material, it's only this kind of this is the only kind of nucleotide that forms genetic material. Okay, so FYI, ATP is also a kind of nucleotide. Okay, so we are going to learn in the future. Okay, the structure of ATP is actually quite similar to the entire to one of the DNA nucleotide. Okay, so I just emphasize again, you have a phosphate group and a pentose sugar and then a base. Okay, so there are 4 kind of bases. So this base can differ from one another. Actually, if you look at this here, so here are the 4 bases. Okay, so we normally use AGTC to represent. Okay, and in the case of DNA, there are AGTC, and then in the case of RNA, there will be AUGC. We'll come to see in a minute. Okay, so pentose sugar, okay, can be, but of course RNA, DNA differ in terms of the pentose sugar. Okay, so for DNA is a deoxyribose sugar and the RNA is a ribo. So actually just compare this. Okay, on the 2nd carbon, okay, so you do see that they differ by in terms of the oxygen. Okay, so DNA, there is a missing oxygen. So the hydroxyl group here is just a hydrogen. Okay, so that's where they differ. Okay, so that's one part where DNA and RNA differ. And it is this difference that decides the difference in name. Okay. And then you have a nitrogenous base. Okay, so nitrogenous base can again be categorized into two kinds. Okay, so there are so we have purine and pyrimidine. Okay, so purine typically are drawn with two rings. Okay, like a hexagon joined with the pentagon. Okay, maybe you should draw this. Okay, or maybe it's up to you. Okay, so because we have it up there as well. Okay, pyrimidine is okay, it's just one ring. Okay, so it's a ring of carbon. Okay, so that's your pyrimidine. Okay, so and then there are two kind of purine, which are Ag and then Tc are the pyrimidine. Okay, so just some if you are supposed to remember which nitrogenous base corresponds to which type. Okay, whether it is a purine or pyrimidine. Because I used to just remember them AgTc. So these two are the purine, and then these two are the pyrimidine. Okay, and the way I remember it is that, okay, the one with the shorter name has larger size. Okay, the one with the longer name, has a smaller size. Okay, that's how I remember them. Okay. Okay, so they are the nitrogenous base, and they are what dictates the difference in identity. Okay, so because the skeleton, so this part is always the same for all DNA nucleotides. They only differ in terms of the nitrogenous base. Okay, so the A stands for adenine, guanine. Okay, and then T, okay, would be thymine, cytosine, and then in RNA later we're going to see they don't have thymine, they only have uracil. So that's another nitrogenous base. Okay, so in DNA we have AGTC, in RNA we have AGUC. Okay, so like I mentioned just now, RNA they don't have a thymine nucleotide. Okay, so only uracil nucleotide, but they both have adenine, guanine, and cytosine. But of course, they differ in the sugar. Okay, and then a phosphate group. Okay, so it's important to know that it is this AGTC that serve as information storage. Okay, for example, do you know how our computer stores information, like when you text or whatever, how is information stored in electronics devices, or when you transmit information, how do you transmit them? How is information stored? Don't know. In Sabah Chinese 有人资讯工艺。Okay, so let's say, if let's say we are in two opposite buildings, okay, there is no phone, you're given a torchlight, okay, and then you want to communicate with each other. How do you communicate? Okay, so, or maybe, sorry? Yeah, so you need code. Okay, so always for information storage you need code. Or if you have watched, all those, especially during that World War, all those decoding Morse code, for example. Okay, so like you can do tab or whatever. Okay, so we can actually use this kind of very simple information. Okay, let's say that just now I say, there's a torchlight. Okay, or maybe or when I say I turn on the light, okay, maybe on off means A, okay, or on off means B or whatever. Okay, so you can actually store information in that format. Okay, so just on and off. In fact, for our electrical signal, always it's just a 100. In SPM you probably learned that as well. Okay, so there's current, no current, current, no current. Okay, so within let's say okay, so that's the first thing. Okay, so you need to have a basic information. You need either on or off. Okay, so 1 or 0 normally in typically in electronic context. Okay, we always do 1 or 0. Okay, we do 1 or 0. Okay, then the next thing you need to decide how many units. Okay, so what is the length of each unit? Or for example, when I'm trying to communicate with you with a torchlight. Okay, so should I, because maybe you on off for like 16 intervals, so should I interpret that like every 2 or every 4, every 8? Okay, so you need to know, or whether, so when I give you a string of these numbers, okay, Okay, do I read this every 3? Okay, or every 4? Okay, or every 8, for example. Okay, like in our case, in computers, okay, in electronic, it's always 8. Okay, so we always are 8 bytes per alphabet, perhaps. Okay, so that's 8 unit encode for one particular alphabet. Okay, so you need to know. Okay, so if I were to have my basic unit, okay, and then what is the length of each pack of information? Okay, so that's something you need to consider about information transfer. Okay, whether you are trying to, do communicate by tapping, your fingers tapping Morse code, for example, or with a with a torchlight, or in fact with optic fiber. That's how information is transferred. So okay, light, no light, light, no light. Okay, or in all these electronic devices, current, no current, current, no current. Okay, so that's how we store and transfer information. Okay, so same thing in biological case. Okay, we want to transfer this information from you, okay, to your offspring, or from the parents to you. Okay, so how do we store that information? Okay, so instead of using 1 and 0, or light and low light, current or no current, okay, so in the case of biological system, okay, we use this nitrogenous base. Okay, and instead of having just 2, okay, like so what all of this, when we say 1, 0, this is what we call binary system. Okay, so just yes and no. Okay, so in the case of biological system, we have 4. Okay, so that's the first thing. Okay, so we have 4 basic unit. Okay, so we store them in the form of AGTC. Okay, so this would be the basic unit of information. Okay, or this is your 1 byte. Okay, so this is your 1 byte of information. Okay, and then you need to decide what is the length. Okay, so how many nitrogenous base correspond to 1 information package. Okay, do you know in the case of human, for example, or in biological system, what is the length of information? Like, do we read, like, let's say, when you get a DNA sequence, A, G, T, C, C, T, whatever. Okay, but how do you know what? Because if I'm giving you a string of codes, you need to be able to read. How do I read this, right? How long is it? Do you know? How long is the information you need? I'll give you a hint. You learn it in SPM. There's a term called codon. Does it ring any bell? No. Okay, so every 3. Okay, so later we're going to see that every three alphabets, okay, every 3 nitrogenous base would correspond to 1 information unit. Okay, later we're going to see it correspond to 1 amino acid, because like I say, you go from your DNA sequences, give you your protein sequences. So protein is made of amino acid. Okay, so of course that is one part of it. Okay, so because you want to think how mother nature passes information. Okay, so you need to think how to store the information. Okay, and later we are going to see how this information is decoded, because if I'm giving you this string of numbers, do you understand what it is? You probably won't. Okay, so because you need to be able to decode it, you need to have a certain certain device or certain means. Sometimes during wartime, like countries, when they're trying to encrypt the information, okay, so they will have like a what we call a cipher. Okay, so they just try to every day they change the code, or how maybe like today, okay, so 1 or 101 means A, tomorrow it means B, and then the day after it means C. So you need to have a way to decode this information. Did you watch a movie, Imitation Game? No. Oh, that's a very nice movie. It's a real story, and it's about Alan Turing, who we call the father of computer science. Okay? A father of computer science. Okay, anyway, so we'll see how these kind of considerations are being taken care of by the mother nature. Okay, so but at least now I want you to know your information unit, okay, each of them. Okay, so each nitrogenous base has 1 byte of information. Okay, so every 3 of them correspond to 1 information unit, which correspond to amino acid. Okay, now I give you mathematical questions. Okay, let me think. Can you please tell me, okay, if it's just 1 and 0, if I'm releasing only 1 and 0, if I want to encode all 26 alphabets, how many of this number do you need? So what would be my minimum length? Okay, let's say if I say, is it possible with just let's just call 2 units, can that encode for 26 alphabets?
No.
No. Okay. 3, can you? No. 4? Or maybe you tell me how many units is required to at least encode A to Z. Let's think. This is your mathematics, your statistics, your permutation, for example.
Permutation and combination.
Just permutation, actually, no permutation, no need for yeah. You probably don't need a calculator. Okay, so just one simple question. If I give you just 2, how many codes can I have? Just 2 units. How many codes can I have?
0, 0,
1. And also?
1, 1, 0, 0.
So 4. Okay, how did you get 4? Okay, so if it's 3, how many? Okay, so for each, so each position, how many possibilities are there? How many choices do you have? At position 1, how many choices do you have? 2. 1 and 0, 2, 2. Okay, so now tell me what is the minimum number of units, okay, to encode just for A to Z? How many alphabets are there? 26. So do you think 4 is enough? No. 5? Yes. Okay. Yes, because 2 power 5 equals to 32. Okay, so with at least in case of A to Z, if you don't even distinguish between alphabet small letter and capital letter, okay, then 5 is enough. Okay, but of course you need to consider in computer, other than this, you have maybe even for capital and small letter, different codes. Okay, and then you have number, you have all the all the comma, whatever it is. Okay, all the punctuations. Okay, that's why you need much. Okay, for example, not typically it's 8 bytes for one information bit. Okay, so same thing. So now for the case of human, okay, let's think. Okay, actually throughout the mother nature, so typically we have about like 20 something amino acid. Okay, so we have the bases. Okay, we have the 4 bases. Okay, AGTC. What do you think is the length of information unit required to store like 20 plus amino acid?
Four choices.
I have 20 plus amino acids. Is two nitrogenous base enough? No. Okay, because if it's just 2, yeah. Okay, so 3 would give you 64. Okay, then we have enough to cover all 20 plus amino acid. Okay, so which is why just now I mentioned. Okay, so the information bit, okay, for in the case of biological system is 3. Okay, so every 3 nitrogenous bases is 1 codon. Okay, so you just need to think, if you are the mother nature or the god, you want to design a biological system. So these are the things you need to consider. How you pass on information. Okay, so yep. Okay, so let's just okay, so you can take this down. Oh. Okay, so formation of nucleotide. Okay, so all of them get like I think I mentioned before. You know, most of the biological molecules are assembled through condensation reactions. Okay, we have learned that whether you have your glucose strung together to form cellulose, or whether it is in the case of amino acid forming proteins or fatty acid reacting with glycerol to form glyceride. All of these are condensation reaction where there is a one water molecule being condensed. Okay, so same thing here. So in the case of the nucleotide formation, okay, you have a phosphate group undergoing a condensation reaction with the ribose, which ribose sugar also undergo condensation with a base, okay, to form, actually no. So this is the part, okay, so where condensation occurs, okay? And just 1 minute. Yeah. Okay, and then they'll be linked together. Okay, so essentially there are there will be two water molecules being released here. It's between hydroxyl group and hydroxyl group, but this is hydroxyl and hydrogen. Okay, but in most other cases that you have seen so far is between like OH and OH. Okay, so just FYI. So two water molecules are being removed. Okay, and ATP is an atypical nucleotide. Okay, like I say, not all nucleotides are involved in formation of your genetic material. ATP is a special kind of nucleotide, which is used as an energy store. We normally refer to ATP as an energy currency. Okay, so basically everyone can use it as a source of energy. Okay, so with unusual feature, so it's unusual in the sense that it has free phosphate group. Okay, so otherwise, if you ignore this, this is the same. This is basically the same as the RNA nucleotide with nitrogenous base A. Okay, so we normally this one we call them adenosine. Okay, adenine or adenosine. Okay, so this nucleotide, okay, this is actually the same as RNA nucleotide, except that it has two extra phosphate group. Of course, if we compare it to DNA, this is a ribose sugar, not a deoxyribose sugar. So that's another difference. Okay, so it's called adenosine triphosphate. Okay, so this part is adenosine, and then this one is triphosphate. Okay, so monomer. Okay, so your nucleotide forming nucleic acid. Again, later we're going to see that between nucleotide, there is going to be another condensation happening, okay, to form a nucleic acid, like DNA or RNA. Okay, so you can just copy this first. Oh, Okay, so here, if you look at two nucleotide molecule, okay, so this is a, let me see, let me find. Okay, so later we are going to learn that this phosphate group can react with the hydroxyl group of another nucleotide and undergoing condensation reaction. I think, yeah, the image is here. Okay, so look at that in a minute. Okay, so what you get is a phosphodiester bond. Okay, because originally this is, okay, so if you look at this particular one, okay, it has a this is a phosphoester bond. Okay, so when the bond is formed, what you see is a POO. Okay, so maybe let me find the case. So here there is a POO. Okay, so I'm sure, or hopefully you know that in your ester normally, what is a functional group for ester? What is it? COO? Okay, so this is ester. Okay, so this is ester. So POO, we call it a phosphoester bond. Okay, so we call this a phosphoester bond. Okay, so when this phosphate group is linked to two different rival sugar, okay, we call it phosphodiester because if you look into it, it is a P. So it has a, okay, let me see the structure. Okay, so if you look at this, here there is a POO, here there is another POO. Okay, this is a phosphoester bond. This is also a phosphoester bond. So it's called a phosphodiester bond. Okay, so here, if you look at this diagram, okay, so it shows you how two nucleotides react together. So this is one nucleotide, this is another nucleotide. Okay, so this nucleotide here, the phosphate group actually has an OH. Okay, sometimes it's just if it's not deprotonated, as in it doesn't release a H, it should have an OH. Just like if you're familiar now, like HCOOH, for example, this is methanoic acid. So when it loses the hydrogen, okay, then it becomes HCOO minus. Okay, so the phosphate group sometimes, depending on the condition, it may actually what we call deprotonates, which means remove the proton. Okay, whether it deprotonates or not. Okay, so anyway, in this case, you have a hydroxyl group here. So this hydroxyl group would undergo condensation with the hydroxyl group on the sugar. Okay, I hope you remember that. Okay, so sugar molecule monosaccharides are whether glucose or fructose. Okay, they have lots of hydroxyl group. So this hydroxyl group would undergo condensation with each other to form a phosphoester bond. Okay, but of course, originally it already has another phosphoester bond, so we call this a phosphodiester bond. Okay, so when you have multiple nucleotides linked together, we call that the polynucleotides. Okay, just like you have more lots of monosaccharide linked together, you get polysaccharide. Okay, so we call this a backbone. Okay, this is what we call the sugar phosphate backbone. Okay, so the bases are projecting sideways from the sugar. Okay, so later we will see how these bases interact with the bases on another strand. Okay, so the sequence of bases is basically that's the information. Okay, like I told you just now. Okay, the AGTC would be the information storage part of your nucleotide. Okay, so and also it's important for you to know that. Okay, so we said the phosphoester bond link the 5 carbon. Okay, of one the 5 sometimes the 5 carbon to the of one sugar and the 3 carbon of the next. Okay, what it means. Okay, so if you remember, again we number the sugar. Okay, so this is 1 2 3 4 5. Okay, so 1 2 3 4 5. Okay, so the phosphate group is on carbon 5. Okay, phosphate group is on carbon 5. Okay, so on carbon 3 there is a hydroxyl group, so it is a reaction between the the hydroxyl group on carbon 3 and the reaction group and the phosphate group on carbon 5. Okay, so which is why it says it link carbon 3 of one sugar to the carbon 5 of another sugar. Okay, carbon 3 of one to carbon 5 of another sugar. Okay, and also just FYI, sometimes we want to know what is the direction of your nucleotide sequence. Okay, conventionally, we would call one end as a 5' end because you see, with this, okay, so phosphate group is always on carbon 5. Okay, so always you have one end with the phosphate group. And then the other end, there will be hydroxyl group. Okay, so we say this is a 5' end, this is a 3' end. So this dot is a prime. Okay, so 5' end is here where the phosphate group ends. Okay, and then 3' end is the hydroxyl group on carbon. Okay, so the hydroxyl group on carbon 3. Okay, so it gives DNA strand a direction. Even for amino acid like proteins, we will have a like in the future, if you do learn more, okay, so you would learn that protein, for example, what are the functional groups of amino acids? What are the functional group of amino acids?
Our group.
Our group is not a functional group.
There should be-
Oh, our group may contain a function, but what are the main functional group of an amino acids? Amino acids. Okay, so amino group NH2 and acid COH. Okay, so of course, as you learn, if you remember from chapter 2, okay, so the N group or the amino group will react with carboxyl group. Okay, so in between is a condensation between amino group and a carboxyl group. Again, we have a direction there. So certainly one N will have a remaining amino group. The other end will have the remaining carboxyl group. Okay, so when we look at a protein, always, okay, conventionally we will call, okay, so this is the N terminal, this is the C terminal. Okay, because for us to, like, we want to know what is sequence. Okay, at least when we study the protein sequence. Okay, so we need to give it a direction. Okay, so we will call this is the N terminal, this is C terminal. So same thing here with the DNA. We call, this is the 5' end, this is the 3' end. So when we talk about a sequence, okay, we need to know which one is what we call as a first. Okay, so first is where the 5' is found, for example. Okay, so anyway, you don't have to be too concerned about it, but at least I want you to know that there is a 5' end and 3' end. Can you please tell me what is a 5' end? What do you have on a 5' end?
Phosphate group.
Phosphate group, okay. Yes, okay, and then 3' end would have a hydroxyl group. Okay, so now let's look at the structure of a DNA. Okay, so your DNA is made up of two polynucleotide strands lying side by side. I'm sure that you are very familiar with okay, so this kind of impression every time when people talk about DNA, a very common structure that you see, okay, that people draw is this double helix. Okay, so actually it is made up of two strands, okay, lying side by side. Okay, so we have two strands of DNA. Okay, so there's two strands run in opposite direction, anti-parallel. But opposite direction, okay, like I told you just now, your DNA has a directionality. Okay, so which means we will call one end as being 5', the other end as being 3'. Okay, so when we say it's anti-parallel, it is this. Okay, So you see for one strand, okay, it's let's say start with 5 prime and 3 prime. The opposite side would be a 3 prime, and the opposite of a 5 prime would be of a 3 prime would be a 5 prime. Okay, so this is what we mean by anti-parallel, okay, opposite in direction. Okay, so yeah. Okay, basically anti-parallel means, okay, so you would have the two strands ending with different ends of the DNA strands, okay? So one end with 5 prime, the opposite side end with 3 prime, and vice versa, okay? So the two strands are held by hydrogen bond between bases is very important here, okay? So actually this whole part is very important about the structure of DNA. It's commonly tested, okay? So you need to know the structure of DNA, how it is held, okay? Just like in the case of like protein, we always learn, the secondary structure is maintained by hydrogen bond, tertiary structure by hydrogen bond, maybe like disulfide bond, and as well as hydrophobic interaction and ionic bond, okay? So later we're going to see how the two strands are held by hydrogen bond, okay? So one thing to know is that your AT, which is your okay, so as we learn you probably already know, okay, so your DNA bases, they are paired, they occur in pair, okay, so A is always paired with a T, G is always paired with a C in the case of DNA, okay, so A is always paired with T, G is always paired with C, okay, so we call this complementary base pairing, and in fact, this is a very, very important feature of your DNA, okay, and in fact for RNA as well, okay. So it is this feature that ensure that later we'll see when the DNA is being copied, you always get the identical daughter. So when DNA replication occur, okay, it is actually due to this particular feature that ensures every time you copy, you get the same DNA molecules being produced, okay? And also it maintain a constant weight, okay? If you note, okay, so A is a purine, T is a pyrimidine, okay? G purine, this is pyrimidine, okay? So if you remember just now, okay, so your purine has 2 ring, pyrimidine 1 ring, okay? So always you have a 2 ring and 1 ring or 2 ring, okay? So which means always you will have a constant width for your DNA strand, okay? Because if you just randomly pair purine with purine or pyrimidine with pyrimidine, okay, sometimes what you are going to get is that you will have a very crooked structure, okay? Some parts are narrower, some parts are wider, okay? So it's not something. So for something that has an irregular shape, it tends to be unstable, okay? Our DNA molecule has to be very, very stable, okay? DNA is actually one of the most stable molecule in our body, okay? So which is why, you know, for bones or whatever that okay, so some of the animal remains dug from the ground from like I don't know few hundred thousand years ago, we may still get some pieces of the DNA because DNA is a very, very, very stable structure, okay? In fact, there are people trying to use DNA as a raw material for making synthetic fiber, okay? Because again, it can be held together by a very strong hydrogen bond, okay? So in any case, so this complementary base pairing case, so later we'll see how it is central to DNA replication. Okay, so of course, structurally, it also has some benefit in that it ensures that throughout your whole DNA, the bond, the width of the DNA is the same. Okay, so the two strands are twisted to form a double helix. Okay, so like I mentioned just now. Okay, so this is the double helix. Okay, and many hydrogen bonds make them a very stable molecule because hydrogen bond is a strong intermolecular force, especially when you have many of them there, then you can hold the two strands quite tightly together. Okay, but of course, when needed, they can also be broken very easily by means of enzyme. Okay, so when we need to replicate the DNA or transcription, perform transcription, those parts can be separate. Those two strands can be separated temporarily. Okay, so they are strong enough to hold it together, but not so strong that they cannot be separated. Because if you have like a covalent bond linking the two, then it's very difficult to separate them. Okay. So Chargaff's rule, okay, so basically it means number of nucleotide with purine equals to number of nucleotide with pyrimidine. What it means, okay, so for example, okay, as you mentioned, okay, your A is always paired with G, C is always A is always paired with T, G is always paired with C. Okay, so let's say in a DNA molecule, you have 20% of T, of A, how many percent of T do you expect to get? Or maybe put this in, just go with number. If I have a 1000 A, then I will get how many? 1000 T. Okay. If I have a 3000 G, I will have 3000 C. Okay. So if you look at this, these two are the purine. Okay. So I will have 4000 purine and you have 4000 pyrimidine. Okay. So that's what we mean by Chargaff rule. Okay. So how many? Okay. So your number of purine and number of pyrimidine will always be the same. Okay. They will always be the same, whatever the identity, whether it is an AG or TC, but the total number of purine will be equal to the total number of pyrimidine. Okay. And of course, we will also know that the percentage proportion of A will be equal to the proportion of T, and the proportion of G will be equal to the proportion of C. If you look at this, for example, okay, you have 12.5% of A, 12.5% of T, and then 37.5% of G, and 37.5% of C. Okay, so one completon of a double helix is about 10 base pair. Okay, so distance of 3.4 nanometer. Okay, so here this is just okay. So here, this diagram, one key thing to note is that okay, your between A and D, between A and T, you have 2 hydrogen bond. Okay, G and C, you have 3 hydrogen bond. This is a very important feature to remember as well. Okay, so AT, you have 2 hydrogen bond. GC, you have 3 hydrogen bond between them. Okay. Okay, so we'll take a short break here, then we'll continue. Okay. Okay, next we'll look at, okay, maybe you try some of the questions. Okay, so and then there's a few questions on the next page. Okay, give you a few minutes to do that, and then I will discuss in a minute. So can you please tell me, okay, what is it? What would be the percentage of bases in the DNA that would be guanine when 38% are adenine? Yeah, okay. Sorry. How much?
38.
No, 12%. Okay, because if you have 38% guanine, okay, so your thymine, also sorry, this is adenine. Adenine would pair with the thymine, okay? So thymine would also be 38%. Okay, so and then in that case, that would be a total of 76%. So the remainder 24% would be guanine and cytosine. Okay, so and then you divide that by 2, then you get 12%. Okay, so the sample has a lower melting. Okay, so this sample has a lower melting point than a sample with only 18% of adenine. Why do you think that's the case? So now this sample that we have has 38% adenine. Okay, so its melting point is lower than a sample with 18% adenine. Why is that the case? Okay, so first, okay, can you please tell me in this case? Okay, so it was 18% adenine. Okay, so the 18% adenine and thymine. What is the percentage for each of guanine and cytosine? 32. Okay. Yes. Okay. So can you please next tell me why this affects the melting point? Oh, by the way, here by melting point, it means the separation of the 2 strands. Okay. What is it? Okay. So melting point here refers to the temperature where the 2 strand is separated from each other. Why do you think that's the case? Okay, so it's because of hydrogen bond. Okay, so can you please be more specific? How would you explain the difference in results? Cytosine has 3 hydrogen bond. Okay, so here when you have a higher percentage of a GC, it means you have a higher percentage of nitrogenous forming 3 hydrogen bond instead of 2. So because you have 1 hydrogen bond, so higher melting and boiling point. Okay, so so the second sample has a higher proportion of okay, I think I'll just rewrite this answer for you. Okay, but okay, the second oh, is it not? Oh. Okay, sorry. Okay, so the second sample has a higher percentage of GC, which form 3 hydrogen bonds. Okay. Just 1 minute. Just 1 minute. So it's being recorded. Higher percentage of GC. Okay.
Has more hydrogen bond is the lower melting point.
No. Okay, so the second sample has a higher melting point. So I'm actually the second sample has higher melting point. Yeah, okay. Can you please draw a box around a nucleotide? Okay, so maybe you can, if you're not certain, you can draw with a pencil and then you can show me and I'll let you know whether you have drawn it correctly. This one. Have you drawn your box? Draw a box around a nucleotide. Can you please draw around a box around a nucleotide? Have you drawn? Around it on the diagram. Okay, so let's have a look. Draw a box around a nucleotide.
Is it a round thing?
Okay, so that looks fine to me. That's okay. No, so because when you draw that, your nucleotide doesn't include the base. Your nucleotide has a base, has a phosphate group, has a sugar. So as long as your box include, okay, as long as your box includes okay, something like this, okay, a base, a phosphate, and a sugar, that will be fine. Okay, or you can draw it like this. Okay, so basically, as long as your box includes a base, a sugar, and a phosphate group, that would be a nucleotide. Okay, label a hydrogen bond. Hydrogen bond should be easily labeled. Okay, so hydrogen bond would be those between the bases. Okay, so the dotted line would be a hydrogen bond. Okay, label a sugar. Okay, so this would be the sugar. Okay, the pentagon would be the sugar. Okay, identify and label the bases A, T, C, and G. How do we know which is A, which is T, which is C, and which is G? Okay, so first of all, if you look at those with 3 hydrogen bond, Okay, those would be the GC. Okay, those with 3 hydrogen bond would be GC. Okay, and of course, G is a purine with 2 ring, C is 1 ring. So this is G, this is C. Okay, and those with 2 hydrogen bond would be A and T. Okay, so A would be the A would be the purine with 2 rings. Okay, T would be the thymine with 1 ring. Okay, so that is how you identify AGTCK. So first, based on whether they have whether they form 3 or 2 hydrogen bond, you can categorize them into GC and AT. Okay, and if you remember just now I say AGCT, AT are the purine with 2 rings, CT are pyrimidine with 1 ring. Then you can actually essentially identify all 4 of them in this diagram. Okay. For example, if you look at this again, this is an here the second part, the second set of nucleotide here. So 2 nucleotide, this is A, this is T. Okay, and then this is G, this is C. If you want to label all of them, okay, so that's how you would do it. Okay, so next, the importance of base pairing and hydrogen bonding. Okay, so base pairing is something that is central to the function of your DNA, or how it is being maintained and passed down to the next generation.
nó không nó không còn nhà cái không nghe cái là nhận cần học từ xe hay brown ôi nó mà gì đó
Okay, so just now we've already mentioned that your the bases, okay, they're pair up A with T, G, and C. Okay, so we call this we describe such a property as being complementary. Okay, so they are complementary or with each other. A always with C always with G. Okay, for example. Okay, so each strand would act as a template. Okay, so template basically refer to something. You know what is a template, right? So basically, it's a basis to determine the sequence of another strand. Okay, so each strand acts as a template. Okay, original DNA gives rise to two copies. We'll look at it later in DNA replication. How this base pairing is very important. Okay, so base pairing is a very important feature under DNA replication. Actually, as shown in this diagram. Okay, so this is the original strand. Okay, so during DNA replication, these two strands will be separated from each other. Okay, so there will be DNA polymerase and enzyme that forms another strand by reading this. Okay, when it reads T, it will bring an A in. When it reads A, it will bring a T in. Okay, so in that case, okay, so by complementary base pairing, you form a new DNA double strand, and this double strand would have the exact same sequence as the parental strands. Okay, so of course, same for the other strand. Again, you synthesize, okay, using one strand as a template. Okay, and because of complementary base pairing, okay, both of the resulting DNA molecules, okay, are identical in sequence to the original strand. Okay, so secondly, in DNA repair. Okay, so again, for example, sometimes you may have a missing, okay, let's say there is a missing nucleotide, perhaps because it is broken here, you miss something. Okay, and then you can refer to the other. Okay, so in that case, you can actually refer to okay, so let's say if this sequence, something happened to your DNA, okay, one of the strand of the one of the nucleotide is broken off. Okay, so you need to repair this DNA. Okay, so by having, okay, by referring to the other strand, okay, then we know, oh, this is a G, so we put a G back in. Okay, because without, if it's not for this particular feature, you wouldn't know what nucleotide to incorporate. Okay, if the DNA simply incorporate any nucleotide into it, that would amount to a mutation where you're going to change the DNA sequence, which is not what we want. Okay, so again, this is the importance of DNA this base pairing, okay, where base pairing makes DNA repair possible. Okay, so such that after the repair you get the same DNA sequence. So that's why we say ensure the integrity of DNA sequence. By integrity of DNA sequence, it means, okay, always we will have the same DNA sequence. Okay, and then of course maintain stability. That's mainly by a hydrogen bond. Okay, so of course, so first your purine pair with pyrimidine, you have a constant width. Like I mentioned, that constant width gives it a very regular structure. Something that's regular is always more stable. Okay, and then hydrogen bond between base pairs. Okay, sometimes you may also have a hydrophobic interaction between stacked bases. Okay, what it means by stacked bases is here. So between the bases here, okay, between the bases here, you have hydrophobic interaction. Okay, so between the bases, you have hydrophobic interaction. So that's everything held together quite strongly. Okay, and of course, between your adjacent nucleotide, your phosphodiester bond, which is a covalent bond. Of course, as you know, covalent bond is a very strong bond. Okay, so all of these contribute to a very stable double helical structure. Okay, so next, okay, so the structure of RNA. Okay, so you can copy this. Yes. 1 minute. Okay, so RNA is very similar in structure to your DNA, except that it is single-stranded, okay? And then it has a ribose instead of deoxyribose. And the other difference being, instead of having thymine as a nitrogenous base, it has uracil instead of thymine, okay? So that's another thing to note. It is typically synthesized in the nucleus, okay? So there are three forms of RNA. Okay, so the mRNA, tRNA, and then ribosomal RNA. Okay, so mRNA, I'm sure it is a term that you have heard, okay, over the COVID period at least, even if you didn't in the past, okay. So there's a messenger RNA, okay, and then there's transfer RNA and ribosomal RNA, which we look at each individually later, but just FYI, rRNA is the one that forms ribosome, okay. So actually we have actually learned in the topic 1 that, okay, so ribosome is actually, so we consider as a cellular organelle for synthesis of protein, okay. So all of these are involved in protein synthesis, okay. So ribosomal RNA is a structural part of your RNA, of your ribosomes, okay. So mRNA forms about 3 to 5% of total RNA, okay. So they convey message from nucleus to ribosomes, okay. So this is something that we'll look at as a process later, okay. So the sequence of an mRNA, okay. So the sequence of mRNA is actually complementary to DNA. So just now we have mentioned that, okay, so every triplet codes for 1 amino acids, okay. So every triplet, okay, every 3 of them code for amino acids, okay. So every 3 of them, we call them the codon, okay. So of course these the codons on your mRNA is going to specify the order of amino acid, like whether the first one is, let's say if you know some of the name of amino acid, okay, maybe the first one will always be methionine, and then maybe tyrosine, and then glycine. Glycine is one that you must have learned, or hopefully you remember before, okay. So glycine is the smallest amino acid, where the R group is a H atom, okay. So basically your sequence of mRNA or sequence of bases on mRNA ultimately is going to specify the sequence of amino acids of a polypeptide, okay. So ribosomal RNA, they made up most of the RNA, 80% of them, okay. So they are synthesized under the control of nucleolus, okay. So, and then they will combine with the proteins to form ribosomes. If you remember, we did mention before, okay, so ribosome is partly synthesized at the nucleolus, okay. So nucleolus is where ribosomes are synthesized, okay. But of course, the first thing that they synthesize there are the ribosomal RNA, okay. So ribosomal RNA combined with proteins to form ribosomes, okay. So we consider ribosomal RNA as a structure because it doesn't really, it's not an enzyme. It doesn't carry out any catalytic function, or it is actually not an information storage unit, okay. So mRNA is messenger, right? So it's the one that relay information, but ribosomal, as the term implies, simply form part of ribosomes, okay. And then we have tRNA. So tRNA is okay, so it has this typical shape that we describe as a clover shape, okay. So this shape that you see is a clover shape tRNA. So it made up about 15% of total RNA, about 80 nucleotide longs, okay. So this transfer also indicates its function, okay. So its function is to transfer things, okay. So so although your tRNA is single-stranded, okay, so it is single-stranded, but it will fold over itself, okay. So part of it would be, form this so you can imagine that some part of this, okay, would be complementary. That's why they can pair up together, okay. Are you with me? Okay, so some part of it would be paired together, okay. So and then at one end of this, okay, so at the 3' end, that's where the amino acid will be attached, okay. So later we are going to see, okay, transfer RNA are called transfer RNA because their function is to transfer amino acids, okay. They transfer it to the ribosomes, okay. So they play a role in protein synthesis, okay. And then of course different amino acids can be attached to this tRNA. Later we are going to see in a bit, okay. So the 3' ends are with CCA, and then there's an amino acid attached there. Okay. So 5' end and end with the G. Okay, there's always the G on the 5' end. Okay, and most importantly, there are 3 codons here. Okay, so there are 3 bases which we call the anticodon. Okay, so there will be 3 bases which we call anticodon, and different tRNA would have a different anticodon there. Okay, so it is actually something very important. So so codon, just now, where do you find codon? Codon. Yep, so on mRNA. So the triplet of codes on mRNA we call codon. When they are on DNA, we don't call codon. Okay, so codon is referring specifically to the 3 codes on an mRNA. Okay, so tRNA is an anticodon, so they are complementary to each other. So if you have a codon for AUC, then the anticodon A pair with U pair with A, C pair with G. Okay, so the anticodon for codon AUC will be UAG. Okay, so later we are going to see that this anticodon is going to pair with the codon, okay, and with the respective amino acid. Okay, so we'll talk about that later. We'll see how this whole design of tRNA actually plays a very important role in converting the code in your DNA, or maybe you can say on the mRNA, into a polypeptide chain, okay, into a sequence of amino acids. Okay, can you maybe just try to fill in some of the differences in the table, okay, based on what you've learned? Okay, can you please just try to complete the table below? Okay, so have you got that? Let's have a look. Okay, so the basic unit of DNA, we call them the deoxyribonucleotide, and then RNA, we call them the ribonucleotide. As we mentioned just now, nucleotide is the entire combination of your phosphate group, sugar, and also a base. Okay, so sugar unit is called deoxy. Okay, the sugar unit in DNA is deoxyribose. Okay, for RNA it is ribose. And then the basis is ATGC on DNA and then AUGC on RNA. Okay. So in terms of structure, DNA is always double-stranded, okay? And then RNA is single-stranded. And there's only one form of DNA, okay? But RNA, we have three different forms, okay? They are all structurally different. Okay? mRNA tend to be just, you know, a floating single strand that has no secondary structure. But tRNA, as you see, it folds upon itself, okay, to form like a clover shape, okay? RNA is actually, you know, incorporated in as part of the ribosome. It serves a structural function, okay? So size, DNA is very, very long, okay? So yeah, I think the other day we mentioned already, okay, so if you stretch all the DNA, okay, inside your cell, you know, you can circle the earth several times, okay? So large and very long, okay? And then what your RNA tend to be very short, okay? So because later we will see mRNA, for example, is more or less the length of a gene or maybe slightly longer than that. Okay, tRNA 18 nucleotides, okay? RNA also is limited in length, but DNA, as you know, DNA can be very, very long where you have like thousands of genes on one DNA molecule, okay? So location wise, okay, so DNA are only found in nucleus. In the case of eukaryotes, of course, they can also be found in the your mitochondria and chloroplast, okay, in plant, okay. And then RNA can be found anywhere, okay. So nucleus, that's where some of the RNA, that's where RNA are synthesized, okay. And after synthesis, they very often go to the cytoplasm, whether it is tRNA, mRNA, or even the ribosomal RNA. Okay, and then ER, we'll see why they are there later. Okay, so in terms of amount, okay, for DNA is always constant, okay? Always, only when there is a cell division, only for cells which prepare to divide, and only before they complete division, okay, you have double amount, okay? So that's only during interphase, okay? But of course, in the case of gametes reproductive cell, okay, they have half of the DNA amount, but otherwise they're always constant, okay? But for mRNA or tRNA, it varies in amount, okay? It depends on whether a particular X cells is actively synthesizing protein or not, okay? So if they synthesize protein, they have lots of tRNA, mRNA, and also RNA, because all of these are involved in protein synthesis. For cells that do not really synthesize, such as synthesize proteins, such red blood cell, such as mature red blood cell, they do not have much mRNA or tRNA or RNA, in fact. Okay, and then stability wise, DNA is super duper stable. Okay, so because they are double strand. Okay, so these two strands, they are hydrogen held together by hydrogen bond. So this strand, so these nitrogenous base are not exposed, are protected. Information is very well stored. So which is why I told you just now that your DNA can be, you know, kept for thousands or even tens of thousands of years underground. Okay, but RNA is very unstable. They tend to be degraded very easily, which is why you try to remember. Okay, so if you get mRNA vaccine, you probably know that they are normally stored in ice, and in fact, they have to be stored in under -20 or even -80 degrees Celsius, depending on the type of RNA or how it is being the solution that they are kept in. Okay, but otherwise, RNA are generally quite unstable. They can be degraded. Okay, too much heat or whatever can damage them and cause them to be broken. Okay, so just this. Okay, so we'll see why DNA is a ideal molecule of inheritance. Okay, so at least in the case of eukaryotes, okay, all eukaryotes use DNA. Okay, for prokaryotes like bacteria, they mostly use DNA as well, actually. Okay, but like virus, they may use RNA as a hereditary material. Okay, so we'll see how DNA is ideal for this case. So first, okay, so carrying information to allow proteins to be synthesized. Okay, so like we mentioned just now, DNA, okay, is what? So sequence of DNA give you gene. Gene is a sequence of gene refer to a unit of information, okay, that is made up of DNA, okay. So of course, ultimately, gene codes for proteins, okay. So DNA carries information for how protein should be synthesized, okay. So the expression to obtain the okay, so they can be expressed, which means, okay, later we'll see how this DNA information, okay, whatever that's stored in the nitrogenous base, how it is going to ultimately lead to production of proteins, okay. We'll see that how, okay, so but basically we say it is being expressed very stable, okay. So two reasons, okay. So first, at least within one strand, they are linked together by phosphodiester bond, which is a covalent bond, which is a very strong bond. So for that reason, it is stable. Secondly, you have two strands, okay, held together by very strong hydrogen bonds, again, that keeps them very stable. So fourthly, it is very faithful in replication. Okay, so when we say faithful, what do you think this faithful means? What does faithful means here? Okay, so if I give you a dictionary, if you copy it, okay, and then you get it 100% correct, no typographical error, that is being very faithful. Okay, by faithful, it means always very, very accurate. Okay, very accurate. So which is why we have a term like integrity of your sequence. Okay, so the faithful replication. Okay, so every time when DNA is replicated, it can be very faithful in the sense that you always okay, you will always, most of the time, you get this correct same DNA molecule being synthesized. Why? Because of the complementary nature. Okay, so because the bases, air will always be paired with T, G always paired with C. And for this reason, okay, so even when the two strands separate and then get replicated, as in they get copied, okay, they will always give rise to the same DNA molecules. Okay, so due to complementary nature. And of course, in any case, like I said just now, I said most of the time it will give you the exact same sequence, because still mistake can happen. If I were to ask you to copy a dictionary, okay, no matter how careful you are bound to make certain mistake. Okay, sometimes those mistakes can give rise to mutation. Mutation is actually something very important. Is okay. So for a species to evolve, we rely on mutation, on genetic variation. Okay, so of course it's something that we'll learn more in depth in the future. Okay, so we'll just finish here today, and then we'll continue tomorrow. Okay.